Hello everyone, hope you all are doing well. So today we are going to discuss the next answer copy of our Aspire for Agriculture test series. So we all know that we had Aspire for Agriculture test series where many students participated, and we are discussing a single good copy of this test so that you can analyze the copies, you can learn from the mistakes of the students, you can learn how to structure the questions, you can learn how to write introduction, conclusion, and all these things can be very helpful to you. And I will be discussing the structure and important points of the question only because reading line by line will take a lot of time. So if you want to read the answers in detail, you can simply pause and read it. Actually, this is of test 7. We have already discussed till test 7 section A. This will be test 7 section B, which will comprise of section of cell biology. So question in this section B, you will be seeing about cell biology. So let's start the answer. So first question was, why is cell called structural and functional unit of life? Okay, so in the introduction part, the student has written about cell. The cell is originated from Latin word means a small compartment discovered by Robert Hooke, so we all know it. Now, why it is called the structural unit of life? Because every organism is made up of cell. Obviously, it is the smallest unit of life. Cells can combine to form new tissues, organs, and systems. Okay. Cells have the power to build up entire organisms. So these are structural unit. Now are they are for functional unit because they perform basic life processes. Okay. In unicellular organism, a single cell is able to do all the vital life functions. Multicellular organism, specialized cells combine and form various organisms. So you all know it. You can simply pause and read it. And well written and a very well diagram of cell with uh, uh, good highlighting of all the organs have been done, well labeled diagram. So that is very good answer. So this was the first question. Now let's move to the second question. Let me make it a bit bigger. Yes. So now let's see the second question. The second question is, write a note on law of independent assortment. So you all know that there are various laws of Mendel, that is law of segregation, law of independent assortment, law of dominance. This one has not written it, but should be written. So these are the three laws of Mendel's. And today we have to, in the question we have to discuss about law of independent assortment. Okay. So what it says that genes and their corresponding segregate independently. Okay. Whenever there are certain kind of genes, they segregate independently during sexual reproduction. Okay. That is, they are not dependent on each other during segregation. They are segregating independently, and then they are getting in the further processes. So the law have been mentioned, and the most important thing which I must say you should write, after writing a paragraph about law, just draw this. Okay, that is showing this phenotypic ratio 9 ratio 3 ratio 3 ratio 1. If you want, you can write genotypic ratio also, but phenotypic ratio and showing this diagram. So it will solve your all problems in this law. So very well written answer. Now let's move to the next question. The next question is, discuss the application of polyploidy. So what is polyploidy? It is the presence of more than two sets of chromosomes in the cells in organisms. Polyploidy can occur into multiple series of 3, 4, 5, 6, 7, 8. That is more than two. So it could be triploidy, tetraploidy, pentaploidy, hexaploidy, depending on the number of series. And now you can see what happened. The question is applications of polyploidy. And it is a 10 marker question. That is, you have to finish in 150 words and around. What student has done? The student has mentioned about types. Autoploidy, polyploidy, allopolyploidy. So it is not needed. If the question is application of polyploidy, just mention about polyploidy. If you want to write the types, you have written already. No need to explain these types. And just move to the applications. So that those things could be kept in your mind to conserve the time also, because writing too much will not give you too much marks. You have to finish the answers in time also. So keep these things in mind. Applications are obviously they are useful in studying the effects of loss and gain of chromosome. Obviously useful in locating the linkages, study of homologous features, identification of chromosome during translocation, production of substitution lines. So well written points. Examples of triticale has been mentioned, that is through crossing wheat and rye. Examples of wheat and cotton have been mentioned, so fairly written. If you want to read it, you can simply pause and read it. Now let's move to the next question. The next question is discuss the measures to overcome self-incompatibility. So we all know there are two terms, self-incompatibility and male sterility. So all these things should be, these two are very important. Either a question for self-incompatibility and male sterility will be there in your examination. So please be prepared. So self-incompatibility is the ability of the plant with functional pollen to self seeds. So set seeds when self-pollinated. So here the uh, in self-incompatibility, the plants have functional pollens, but they are not able to set seeds. But in male stability, what happens? The plants doesn't have the functional pollen only. So this is the basic difference. Keep in mind. And again, you said the types of self-incompatibility have been explained, which was not needed. Here the question is directly asking the measures to overcome self-incompatibility. So you should have directly gone to measures. So keep these things in mind. So it can be helpful in doubling chromosome number, isolating fertile mutation, transferring self-compatible some compatibility alleles. Okay, use of chemicals, bud pollination, stop pollination, controlled hybridization. So written points. You can simply pause and read it if you want to. High temperature, that is temperature treatment can be done. In vitro fertilization can be done. So these are the ways to overcome self-incompatibility. Now let's move to the next question. The next question is discuss the characteristics of cytoplasmic inheritance. So we all know what cytoplasmic inheritance is. That is, there are some kind of traits that is inherited. So what are its characteristics? Maternal effects, obviously, non-Mendelian segregation, uniparental inheritance. So written points, you can simply pause and read it. I don't want to go in detail in these things because I have all discussed in the static video lectures, and various times I have solved these types of questions in the PYQ sections. Okay, because all this has been asked several times in PYQs. Now let's move to the next question. The next question is discuss the various phases of cell cycle. So you can see in the introduction the student has made this diagram which enhances your answer. So the phases have been mentioned here, and we all know that there is G1 phase, S phase, G2 phase, M phase, interphase. So in interphase, there these are all phases. Okay, actually there is two phase, interphase and M phase. So in interphase you can see G1, S and G2, and M phase is different one. So interphase you can simply pause and read it. It is known as resting phase. Then we have G1 phase. So it is initiation of replication happens there. Then we have S phase, here DNA replication takes place. Then G2 phase, here RNA proteins and other things are multiplied. Then M phase where mitotic divisions takes place. And in M phase all the five phases, that is interphase, prophase, metaphase, anaphase, and telophase have been mentioned. So very well written answer. And then cytokinesis, that is cell is divided into two parts. So this was the next question. Now let's move to the next question. The next question was what is heritability? Discuss its types and also discuss its advantages. So heritability is the property of total variance that is attributable to the average effects of genes. That is what determines between relative the degree of resemblance between relatives. So very complicated definition, but well written. I am not saying wrong. Simply keep it in mind that heritability is nothing but hybrid figure. Okay, that is our vigor is better than the parents. This is the most simple thing. Okay, here what are the evolve that heritability is ranges from 0 to 1, so 10 points. Now types have been mentioned, that is broad sense heritability and narrow sense heritability. If you want to read it, you can simply pause and read it. I have discussed all these things, that is why I am not going into detail. I am just giving a structure of the answers. Now, what are the advantages of heritability? Obviously, for understanding complex traits, for predicting, diagnosing, treating diseases, for estimating progress and setbacks on traits, improving crops. So, level written points. You can simply pause and read it. Now, let's move to the next question. The next question is discuss gene pool along with its types. So, gene pool is the complete set of genes and all their different forms. Okay, so genes, their different forms, all the compositions of genes, all this comes under the gene pool only. Now we will see the types of gene pool. So it is, there is primary gene pool, there is secondary gene pool. Okay, there is tertiary gene pool. So if you want to read it, you can simply pause and read it. Okay, line. So, and I have discussed all these things. That is why I am not giving too much effort here. I am just giving an overview. You can read the points, they are well written. So this was about gene pool. Now let's move to the next question. The next question is what is linkage? and discuss its types and briefly explain about detection of linkage. So we all know linkage was discovered by Batman and Punnett, then T.H. Morgan. We all know the fruit fly experiment where the theory of linkage was derived. So now in the question types and detection. So types of linkage on based on crossing over there is complete linkage, that is completely the linkage in on the complete sets. Okay. Then we have incomplete or partial linkage based on gene involved. We have coupling phase, we have repulsion phase. Based on chromosomes involved, we have autosomal linkage, that is other than sex chromosome, sex linkage. So very validating points. Detection, how we can detect the linkage by using chi-square test, very good. We can do recombination frequency test. We can do LOD score. We can do regression analysis. The validating points. You can simply pause and read it. Now let's move to the next question. Next question is what are haploids? Discuss it, production and usage. So haploids, that is name only suggests haploid, that is one set. That is they will have single set of chromosomes. Diploid, that is two. Then polyploid, that is more than two. So we should all keep all these things in mind. Very important ones. Now production of haploids through anther culture, ovarian or ovule culture, delayed pollination, irradiation of pollen, chemical treatment, temperature shocks, all these things can lead to the production of haploids. Now usage for plant breeding, genetic studies, studying chromosome, production of homozygous lines, unmasking recessive gene information, establishing gene map, producing somatic habits. So bulletin points. You can simply pause and read it. Now let's move to the next question. The next question is discuss the function of mitochondria. Also draw a well-level diagram of mitochondria. So we all know mitochondria known as the powerhouse of the cell, has various functions such as respiration, oxidation of carbohydrates so that energy can be produced, dehydrogenation. So related points, it has also some mitochondrial DNA on it. We all know that three parent baby is highly in use, so you should read it for UPSC examinations. Where also there is huge role of mitochondria. They have role in Krebs cycles, although that is animal. We are discussing about agriculture, also that is plants. And well, diagram of mitochondria is made. All the things such as outer membrane, ribosomes, ATP synthesis criteria, crystal DNA, all the things have been well mentioned and labeled. So very well written points. You can simply pause and read it. So this was section B of test 7, and we have just briefly got a overview of this answer paper. So that is all for today. I hope you have read the answers by pausing the video at different intervals. So I hope you like it. So if you like it, please press the like button. Please do share and please do subscribe. It takes a lot of hard work to make such videos for you on regular basis. And if you like, if you share, if you subscribe, it motivates us to make more such videos for you regularly. So that is all for today. Have a nice day. Thank you.